Hello everyone, my name is Skarleth Cardenas. I would like to start by taking the Aging and Rejuvenation Conference for the opportunity to present our work on the positive feedback loop between chronic lysosomal dysfunction and circular RNAs in Alzheimer's disease mouse models. I don't have financial disclosure to report. Alzheimer's disease or AD is the most common form of dementia. It is projected by 2050 approximately 12.7 million people will be living with Alzheimer's disease. This disease is pathologically characterized by the accumulation of amyloid plaques and neurofibrillary tangles. At the molecular level, there are two primary processing pathways for the amyloid precursor protein or APP. In the non-amyloidogenic pathway, ADAM10 cleaves APP to produce soluble APP alpha, which prevents plaque formation. In contrast, in the amyloidogenic pathway, base-1 cleaves APP to generate soluble APP beta, which promotes amyloid plaques formation. Unfortunately, there are still no effective treatments for Alzheimer's disease. Recently, a new class of non-coding RNAs called circRNAs has been identified. These are form back splicing between exons and introns. Circular RNAs are unique in that they lack 5' and 3' ends, giving them a circular structure and protects them from degradation. One of the most important roles that has been described for the circular RNAs is as a microRNAs sponge. Interestingly, microRNAs are known to regulate several molecular pathways in Alzheimer's disease, including base-1 and APP expression, thereby reducing amyloid plaque accumulation. I apologize for this complexity, for the complexity of this slide. In this table, Dove and collaborators identify circular RNAs that are significantly altered in Alzheimer's disease patients compared to healthy controls. On the right, the circular plot shows red dots representing circular RNAs associated with clinical dementia rating, Braak staging, and Alzheimer's disease diagnosis, along with their chromosomal locations. On the other hand, autolysosomal dysfunction has been shown to contribute to Alzheimer's disease pathology by promoting tau tangles formation and A-beta accumulation. This dysfunction is driven by multiple genes involved in the autolysosomal pathways. Interestingly, emergency evidence suggests that circular RNAs can regulate autolysosomal function by sequestering microRNAs. However, the relationship between circular RNAs and lysosomal dysfunction in Alzheimer's disease is still poorly understood. So that our research question was, what is the role of the circular RNAs downstream of chronic lysosomal dysfunction in amyloid beta accumulation in Alzheimer's disease mouse model? Our hypothesis was that chronic lysosomal dysfunction would affect circular RNA levels, increasing amyloid beta levels in the Alzheimer's disease mouse models. To test this, we crossed the 5XFAD, which is an Alzheimer's disease mouse model, with two different lysosomal enzyme deficient mice models. One had 50% less expression of PPT1, which is involved in lipid metabolism, and we call it as a P5X. The other group had 50% less expression of NAGLU, which is involved in carbohydrate metabolism, which we call it as N5X. Both proteins are lysosomal enzymes. And as first results, our first results, we observe reduced survival rates in P5X and M5X animals, which we attribute to lysosomal deficiency. At the top of this slide, we can see representative coronal brain slices from each group, stained to show amyloid plaque accumulation in the black dots or the black signal. At the bottom is a quantification of this amyloid plaque. As we can see, both enzymes deficient groups exhibit a significant increase in amyloid plaques compared to the 5-XFAD controls independently of the lysosomal enzyme deficiency. Additionally, we quantify insoluble amyloid beta 40 and amyloid beta 42. As shown, lysosomal dysfunction significantly increases the accumulation of both fragments in the hippocampus, suggesting an exacerbation of Alzheimer's disease pathology. We then ask which circRNAs are affected by lysosomal enzyme deficiency. Using RNA sequence, we identify differentially expressed circRNAs. The volcano plots show downregulated circRNAs in blue dots and upregulated circRNAs in red dots across both comparisons between 5-XFAD versus P5X and 5-XFAD versus M5X. After that, we classify or we identify the top of the circRNA between comparisons. So upon analyzing circRNA expression data, we identify 6 circRNAs that were significantly upregulated in both comparisons, P5X versus 5-XFAD and M5X versus 5-XFAD. Similar results we observed with the circRNAs that were significantly downregulated across both models. This suggests that chronic lysosomal dysfunction influences circRNA expression in a consistent, directional manner independent of the specific enzymatic deficiency. Next, we perform in silico analysis to identify potential interactions between these circRNAs and the target of microRNAs. And we found that multiple circRNAs can sequester the same microRNA, suggesting that circRNAs may play a central role in fine-tuning Alzheimer's disease-related gene regulation. In conclusion, our results show that chronic lysosomal dysfunction leads to altered circRNA expression, which may contribute to Alzheimer's disease progression by influencing APP metabolism. Let me give you a specific example. In the APP-PS1 mouse model, miR13855P is upregulated. This microRNA suppresses ADAM10 and SIR1, both of which are protective against amyloid beta production. Our in silico analysis suggests that SIRPAN3 and SIRC-ADAM10 can potentially bind and sequester this microRNA. However, in our model, these circRNAs were downregulated, potentially releasing the microRNA 13855P and promoting A-beta accumulation, as we can see in our stain of the slices. This remains a hypothesis and needs experimental validation. For that, such as a future direction include experimental confirmation of the circular RNA microRNA interaction, temporal characterization of the circular RNA expression during AD progression, and ultimately validation in human samples. Finally, I would like to thank my mentor Bruno Benitez for guiding me through this exciting project. Thanks also to my lab mates, Mayu and Matthew Rosin, and to our collaborators at Washington University in St. Louis, and the NIH and NIA for their support. Thank you very much for your attention.